From this lab 1, we can conclude that there are several methods in order to obtain sample and tissue from fish. From dissecting the fish, we can see various techniques to observe their external and internal organs such as gill, liver, kidney, spleen, and many others. Histology is vital to studying the structure of biological tissue as well as the understanding and detection of disease. Although more and more histological staining techniques used for detecting protein, lipids, and nucleic acid have been developed, histological techniques are still required in this field. Histological research has recently developed advanced high-resolution, high-throughput, and 3D histological techniques that will greatly facilitate histological research. However, the new developed technique also face the consequences of extracting conclusions from the plethora of data generated by the use of advanced techniques. In the future, more accurate techniques are needed to achieve accurate and firm results.
For Lab 2, we investigate a massive fish diet virtually. In this lab, we understand the basic concept of sample collection, perform fish necropsy, concept of trophic pyramid, and the energy flow in ecosystem. Not to mention, we also learn the difference between heterotroph and autotroph organisms. As we know, fish have gills that are designed to fill with water and take oxygen from it to breathe. Fish will suffocate if the water lack of oxygen, which usually caused by a variety of factors including harmful algae bloom, overcrowded fish pond, and limited water surface area. As a result, this investigation had been done to prevent those problems. We can avoid fish suffocation by using aeration, as this will circulate the water and raise dissolved oxygen to restore the aquatic environment balance. Next, we can control the growth of algae by simply remove it to clear up the water column. We can also use phosphorus-free fertilizer to limit nutrient-rich runoff that will stimulate algae growth.
Media is very important in the process of studying the microbiological organisms such as bacteria and fungi during their time inside the lab. It is widely used for growth, separation, and purification. This is due to the fact that they could efficiently provide the most suitable condition for the microscopic organism. Aseptic techniques and autoclave are practiced that prevent the contamination of growth media. The only difference between broth and agar media is that broth do not contain an agar component. This simple formula of nutrient agar provides the nutrients needed for the development of a large number of bacteria that are not too fuzzy. In order to prepare media in laboratory, materials such as tryptic soy powder, distilled water, spatula, aluminium flask, screw cap bottle, measuring cylinder, magnetic stirrer, hot plate, petri dish, and autoclave are needed.
Next, I will explain about Lab 4, which is bacterial quantification by culture. The objective of this experiment is to set up a serial dilution of bacterial culture and describe why they are used. In addition, they are also used to calculate the colony forming unit from colonies counted on agar plate based on dilution factor and volume correction factor. In summary, the experiments are done to observe and study the colonies forming abilities of bacterial culture. Serial dilution are useful when we want to obtain a very diluted solution from every concentrated stock solution. This helps us in order to select a specific type of bacterial colony, as many types of bacteria tend to grow in a prepared culture media after putting them under controlled condition. The colonies could be counted by using the formula above. In conclusion, this diffusion is an important step in order for us to identify the rate of colony forming of bacteria inside ideal condition.
In Lab 5, the topic is about the identification, bioassay, and health monitoring of shrimp. To understand the procedure on identify the disease on the shrimp is to discuss common disease infected on shrimp. In this lab, we learned that disease is one of the many factors that contribute to the demise of shrimp stock other than facing the potential of mortality due to old age. Although estimates of the importance of illness in natural populations are frequently incorrect, the impact of disease, predation, and hunger is recognized as critical in limiting the number of natural stock whenever they are expanded to excess. In conclusion, some shrimp-associated organisms occasionally become substantial disease contributors because of high density and confined rearing, which is unnatural man-induced stress. To mitigate the unfavorable outcomes, special precautions are required.